So we left off with metaphase. So now we'll begin part 3 of chapter 12's lecture and PowerPoint. So we left off with metaphase, and again, the key characteristic of metaphase is that the chromosomes are aligned along the middle of the cell. This is a great picture here on the right showing the microtubules and the chromosomes there lining up. And you can also see on that lower picture the kinetochores, they're attached to the mitotic spindle. So next phase we're going to look at is anaphase. So once we get through metaphase, the key characteristics of anaphase include that the sister chromatids are going to be separating at the kinetochores. Another way to say that is that the sister chromatids are going to separate at the centromere. They're going to be moving to opposite poles, so I always think of anaphase as the apart phase. They're pulled apart at the centromeres, and they're actually pulled by motor proteins, those ones that walk along the microtubules. So those are what are going to drag those separated chromatids to opposite ends of the cell. At this point, we need a lot of ATP for those motor proteins to do their work. So the mitochondria are going to be working hard to produce the ATP that is needed by the cell for mitosis. The separation of the chromatids is really important here. So we have the double-stranded chromosome here, which is the two chromatids joined at the centromere. And we want to get those two chromatids separated so that we have two identical chromosomes, one going to each end of the cell. And this is just a picture. Some studies were done to show this movement and how it occurs. So you can see here the depiction of a motor protein and how that is using the microtubule to walk along and pull the chromosome along. So the purple is the chromosome there on the top. The yellow is the part of that mitotic spindle. And what's happening is that microtubule is also shortening in the process, which makes sense when you think of those microtubules as being composed of actin and myosin, similar to our muscles, which are going to shorten and lengthen as well. So our last phase of mitosis is telophase, and the key characteristic that here is to think of the daughter nuclei forming and the chromosomes dispersed to the two separate ends. So the chromosomes have arrived at the opposite poles. We now have the reformation of nuclei. We no longer have the chromosomes visible under a light microscope, which means that that real intense condensing that has occurred is now loosening up again. So the chromosomes are loosening up. We also have the disappearance of the spindle fibers. They're being dismantled. And then we have the beginning of what we call cytokinesis. And some people think of that as not really part of mitosis, that it's just something that occurs after the end of mitosis. But really and truly what you just need to know is that it is required and has to occur. And it occurs differently depending on whether it's an animal cell or a plant cell. So in an animal cell, we have a cleavage furrow occur. And so you see that picture there on the top of a cleavage furrow occurring in the cell. And you can think of it, what's occurring actually is like a constriction, like a belt being tightened. And that belt is made of actin and microfilaments around the equator of a cell. So it's just tightening it off to actually cause the cleavage that's needed in order to get our two daughter cells. This is a picture showing actual mitosis occurring. So you see the different phases depicted here. You see interphase there on the top left, and then prophase, metaphase. You can see anaphase there at the bottom, and then we see telophase, where we have the nucleus being reformed again, and then late telophase, where we actually see a very clear nucleus forming. This is another picture, and this is showing prophase. And prophase, again, what's occurring here, the chromatin are condensing, the nucleolus is disappearing, and the nucleus is starting to disappear as well. In prometaphase, we have the condensation of the chromosomes occurring. They're very visible now. Metaphase, they're lining up in the middle. Anaphase, they're being pulled apart. And then in this case, we have in telophase a cell plate being formed. So this tells us, not only does the shape of the cell tell us that this is not an animal cell, because we actually can see a cell wall, we can tell that this is a plant cell. So in cytokinesis, one of the differences that we're going to see is that a cell plate forms during that telophase. So what a cell plate is vesicles lining up at the equator, and those vesicles have come from the Golgi apparatus. And they fuse to form two cell membranes. And that new cell wall is laid down now between those two membranes. And the new cell wall fuses with an existing cell wall along the edges to form two daughter cells. Here's a picture showing that occur. So you can see that cell plate forming here. Late telophase and cytokinesis in the plant cell. And you can actually see the circles of those vesicles lining up. This is a different slide showing mitosis in the plant cell. So you can see interphase and prophase, prometaphase, metaphase are all looking very similar in animal and plant cells, and in anaphase, very similar. And not until telophase do you see this clear, distinct cell plate form there in the middle. And again, this is showing onion root tip, which is really commonly used as a way to practice identifying the different phases of mitosis. So you might want to just take a minute to look and see how many phases you can identify here. See if you can find one of each of the key phases of mitosis. And if you have trouble, there are plenty of websites online that you can practice and get some help in identifying by a photograph what phase is going on, and you really do need to know how to do that. So now we've looked at what's occurring in eukaryotic cells during mitosis, but we should take a second to think about prokaryotes. Bacteria and archaea reproduce by a type of cell division that we call binary fission. And in binary fission, the chromosomes still replicate. Remember, in a prokaryote, particularly in bacteria, the DNA is in a circular form. And so it has an origin of replication where replication begins. So here's a picture showing binary fission in a bacteria. You can see that top little dot, origin of replication. That chromosome's a double-stranded DNA. When it gets replicated, we end up with 2 circles of DNA. And then with binary fission, it's much more simple. We just have the single circular chromosome separating out to 2 different ends. So we have 1 of each going to each end of the cell. We get a ring of proteins forming in the middle of the cell, which is where we have that fission that's going to occur. And so there's a pinching that occurs between the 2 cells to separate them into 2 new daughter cells, each with a copy of the original bacterial DNA. So there's a thought that all cells have evolved from bacterial cells or prokaryotic cells. So there has been, and if you look at different types of cells from prokaryotes through protists to eukaryotic yeast to eukaryotic animal cells, you can see a lot of similarities in mitosis. And you can see it becoming more complex as the organisms become more complex. So there's a possible progression of mechanisms between binary fission and mitosis that you can observe by looking at these different organisms. So next I want to take a quick look at how cell division is regulated. So cell division is regulated through a molecular control system. The frequency of division is going to vary with the type of cell. As we already saw, some cells never go through cell division, like brain cells, nerve cells. And others are going to have to replicate and divide on a very frequent basis. So these differences have to result from regulation at a molecular level. And it appears to be driven by specific chemical signals that are present in the cytoplasm of the cell. So there are things within the cell itself that are going to regulate how often cell division occurs. And there also are some external factors we'll look at the end. But first, looking at within the cell, there's a cell cycle control system, which you can think of as a clock. So if you think of the cell cycle as a clock, there are specific checkpoints along that rotation of the clock. And if there's not a go-ahead signal at each of those checkpoints, then cell division is not going to occur. So here's a picture showing the 3 of those checkpoints that we know of. There's a G1 that's going to occur in the G1 part of interphase. There's a G2 which has to occur at the end of the G2 phase. And then there's also a mitosis or M checkpoint that occurs in the midst of mitosis. So for many cells, the G1 checkpoint seems to be the most important one. So if a cell receives a go-ahead signal at this G1 checkpoint, it's usually going to complete the rest of the phases, both of interphase, both the S and the G2 phase, and then go into the mitosis phase and complete division. If the cell doesn't receive that go-ahead signal, then it's going to exit the cell cycle and switch into that G0 phase, which is that non-dividing resting phase. So what's controlling this clock are cyclins and cyclin-dependent kinases. So these are both regulatory proteins involved in the cell cycle, and they're closely related, as you can tell by their names. The cyclin-dependent kinases have to have cyclins attached in order for cell division to occur. And so the amount of cyclin present and the cyclin-dependent kinases, or CDKs, fluctuates during the cell cycle. And the maturation promoting factor is actually that combination of a cyclin-dependent kinase complex